Just based on what we've gone through so far, taking a look at the different ways proto-oncogenes can be converted to oncogenes, and then going along with that, what are the treatment options that you have to try and go in and selectively take out cancer cells that are expressing one of those oncogenes. You can see already it's a real mixed bag, okay, in terms of succeeding at that. And the reason it is because, one, the accessibility of the target itself and how amenable the target is to designing a drug, that's a real issue, okay? So, for example, if you have oncogenic forms of receptor tyrosine kinases, you basically have two options there. You can target the outside using, a monoclonal antibody that will block the extracellular region of the receptor from binding to the ligand. You can interfere with dimerization. You also have the option of targeting the tyrosine kinase domain. So you can use inhibitors for that. But then the problem is that, remember, the receptors are the first step in the pathway. So that what you're doing, again, is as soon as you put selective pressure on cells, then you're selecting for them to acquire adaptive mutations that will just allow them to skip or bypass whatever component it is you just took out. So the limitation of targeting receptors is that they're at the very, very top of the pathway, which means you've got a lot of downstream events that still have to happen where you could select for the acquisition of mutations that'll just bypass it, okay? And you saw then that if you do target a receptor, the most logical, the next component that's typically going to be converted to an oncogene is the G protein RAS. And RAS poses a real problem simply because most drugs inhibit a biochemical activity. And the problem with oncogenic RAS is it lacks a biochemical activity that it should have, namely its GTPase activity. So it's very hard, again, to target an enzyme that has a problem because it's not doing what it's supposed to. It's a double negative. And so the one thing that you saw was RAS poses a real therapeutic challenge simply because there isn't much to grab onto, and then secondly, you're really limited in terms of what sort of activities you can take it out, okay? So we're going to continue down the pathway. And what we're going to do is that you're going to take a look and see, we don't have to keep chasing the oncogene every time. And so what you're going to see is we can actually take a little bit of a detour. And that is we can go after a phenotype or a hallmark that a particular type of cancer is manifesting because of the oncogene it has, okay? So, but before we get to that, here's the next guy who's in line after RAS. And again, in the normal RTK signaling pathway, here's where we switch over from the G protein at the plasma membrane to cytosolic kinases, and we're off on this kinase cascade. The kinase is RAF. Now, remember in the slide that we talked about in the normal signaling pathway, the emphasis was RAF is normally inactive because it's bound to a repressor protein 1433. And that RAF has to interact with RAS. God, hitting those two isn't easy. So RAF has to engage with RAS to enable the repressor protein to dissociate. And that's true, okay? But there's another component to that that the textbook doesn't touch on, but this little review article that came out a few years ago actually does, okay? And that is, what they're not showing is the dissociation of 1433. So that's happening, it's just in the background. So if you guys want to draw 1433 bound to the inactive form of RAF, and it's bound to a phosphorylated residue on it, and that's what's repressing it, that's cool, because that's still there. But I want you to focus on something a little different, okay? And that is the binding of the inactive form of RAF to RAS also triggers its dimerization, okay? And you notice that it takes two individual RAFs, each of which binds to a separate RAS, okay? So we got this little cute structure up here. So the interaction between RAF and RAS requires that RAS has GTP bounds. You already know that. But then once we form that homodimer with RAF, it can dissociate from RAS. So it does not have to remain bound to RAS to be catalytically active. It only has to form that homodimer, okay? It's also gotten rid of 1433, so that still applies. So the point is, once it disengages from RAS, it's catalytically active. It is going to phosphorylate the next guy in the pathway, which is MEK. That's the dual specificity kinase that activates MAP kinase, also known as ERK, and then we go down the rest of the pathway. Okay? Now, remember that if you block a receptor, that the most common mechanism of acquiring resistance to an inhibitor of a receptor is to activate the oncogenic form of RAS. But you can also activate an oncogenic form of RAF. Okay? This happens quite often in colon cancer, but it particularly happens in malignant melanoma. So in the most prevalent forms of skin cancer, it turns out RAF is generally the oncogene that gets activated. Just a different cell type, different mechanism, but it's the culprit more often as not. Okay? The most common mutation that occurs in RAF that enables it to become an oncogene is a single amino acid substitution of valine 600 to glutamic acid. Okay? Mimics phosphorylation. Okay? But here's what's interesting about it. Okay? So what happens is this mutant kinase domain now is constitutively active for two reasons. One is it doesn't have to dimerize, and secondly, it doesn't have to go through RAS to become active because it won't bind 1433. Okay? So the V600E mutation prevents it from binding to 1433, so you can't repress it. Secondly, it allows it to become active as a kinase as a monomer. So now we have absolutely no need to go to RAS. Okay? We skip that step. Well, it turns out you can go after, and you could do a high throughput drug screen to find compounds that would inhibit the kinase activity of RAF V600E. And it turns out they did this, so they went through, did a high throughput screen, and they came up with this compound over here on the right, vemurafenib. It's a kinase inhibitor, okay? Very effective, very potent, works quite well. The neat thing about it is vemurafenib is very, very specific for just the V600E allele of RAF. So the nice thing about this is that this illustrates a tumor-specific kinase inhibitor, okay? Because you're not going to find that form of RAF in normal cells. You're going to see that we can take the same approach to knock out another unique oncokinase that occurs in B-cell lymphomas. We'll talk about that in a couple of minutes, which is the drug Gleevec, okay? But anyway, so we go in and we can target RAF using vemurafenib. But as soon as you do that, once again, the cells go to work. You're applying a selective pressure on them, so you're going to be selecting for adaptive mutations that allow you to bypass the drug. They're going to become resistant. Now here's what's interesting about how you acquire resistance in this case. And that is, you actually undergo a deletion of the amino-terminal portion of the RAF kinase. So there's actually a chromosomal deletion. We remove the whole first half of the protein. Now what that does, it eliminates the regulatory domain, it eliminates the 1433 binding domain, but what it does is it now allows RAF, which is normally only active in normal situation, when it dimerizes, okay, this mutant version, the V600E, eliminates that requirement. But now when you delete the N-terminal regulatory domain, you also take out the binding site for the inhibitor. So what it does is it allows the cell to acquire resistance by eliminating the binding site for the inhibitor. And now this truncated form of RAF, which is called P61B, this little truncated stub is now constitutively active. You're left with a kinase domain, no regulatory domain, no inhibitor domain, no nothing, and now this guy can resume signaling. And you have no way of stopping it, okay? So can you appreciate the problem therapeutically that if you always go after the oncogene, the minute you do it, you're applying selective pressure to the cells, they undergo adaptive mutations, and one way or another, they're going to become resistant. The resistance can either come about by activating some downstream target, or it can come about by actually targeting another mutation in the same gene that you just went after, okay? It's the real struggle with cancer in general. Now, you can look at it as a bad news. You can look at it and say, damn, this disease is going to be very, very difficult to treat. We're always going to have to use combinatorial therapies. But on the other hand, look at it from the cell's ability to survive. This is really an incredible testament to our cells' ability to survive. You hit them over the head with a drug, you hit them over the head with 2 drugs, and you notice they will do anything within their grasp to survive, okay? So it kind of speaks well, I guess, to determination and a real desire to survive at all costs. And that's another take-home that you can see from the way that these cancer cells are continually adapting, having these mutations, and they're going to keep on trucking at some level. And that's always going to be the challenge. But what this points out here, what if we move away from this? What if we step away and instead of always trying to target the oncogene, let's target the phenotype that's being driven by this oncogene? And so here's where we can switch a little bit. And the reason we can switch here is because the most common form of colon cancer is driven by mutations that are either in RAS or RAF. Okay? Right here, the G protein that is involved in activation, and then the kinase itself. Okay? So what is the phenotype of these cells that might lend itself to an alternative chemotherapeutic approach? Okay? And here it is. Oh, sorry, guys. Bang, it's Monday morning. Okay, another way, before we get into that, here's another way that we can try and come up with a trick. And the trick here is, RAS is very difficult to target. RAF, as you can see, will become resistant. So here's another paper that just came out about a year ago that says, what about designing a drug that instead of blocking the kinase activity, what we can do is to try and prevent the interaction between RAS and RAF. And what this is a small molecule called rigosertib. And what this does is it actually binds to the surface on RAF that would normally interact with RAS. Okay? And so what this would do is to prevent the oncogenic form from RAS from activating RAF. Okay? This is relatively new. It looks like it's promising on cells in culture, but it is yet to go through clinical trials. So it's kind of like, let's wait and see. But of course, as you guys know, the limitation of this approach is that we can make oncogenic forms of RAF that don't require an interaction with RAS. And so those are going to bypass that particular treatment anyway. Okay, back to the phenotype. Our favorite chemist is back up here again. Okay. And it turns out he might have been right. Okay? So, 50 years ago, back in the late 60s, early 70s, Linus Pauling became a very, very strong, outspoken, almost consigned to the lunatic fringe of science because he was an advocate of megadoses of vitamin C as a key to longevity and, more importantly, as a key to preventing cancer. Okay? And in fact, you can basically buy Dr. Linus Pauling vitamin C, megadoses, and he's not talking about the typical 500 milligrams of vitamin C you'd take, you know, non-prescription over the counter. He's talking megadoses of vitamin C, gram amounts of vitamin C, literally 15, 20 grams a day. You consume that much vitamin C, I can tell you one thing. Your urine is going to be so acidic it will dissolve porcelain. So it's water soluble. You will excrete it. It will do a great job of acidifying your urine. But what you're doing is that vitamin C, as you guys know, is a reducing compound. And the reason that Pauling touted its potential is that they knew back then, going as late 60s, early 70s, that reactive oxygen species can damage cells. They can damage DNA, they can damage proteins. It's involved in cancer, it's involved in aging. So Pauling was an advocate of these megadoses of vitamin C to basically use it to try and ward off cancer. Okay? Now, as we talked about it, Pauling lived into his mid-late 90s, so maybe it worked in his case. But basically, the idea of vitamin C megadoses was just kind of like, oh, come on. Where he had it wrong was in the idea that it might prevent cancer. That's not the same thing that it may not work to treat a particular type of cancer. And it looks like it might be the case. but it's doing so in a way that Pauling never would have envisioned. Okay? So we're looking at colon cancers that are being driven here by the RAS or RAF oncogenes. These are transgenic mice that have basically been engineered to express either a RAS-D allele or a dominant RAF V600E allele. Okay? These mice develop colon cancer at a very, very high frequency. Okay? And this is work that was done in Lou Cantley's lab, and he's one of the premier cancer researchers out there in the world. Here's what they noticed. You look at these cells that are overexpressed, or not overexpressing, but they are expressing these mutant oncogenic forms of RAS and RAF. What are the hallmarks of these cells? Is there something else we can go after besides targeting the oncogene? What they noticed are two things. One of the first hallmarks is that as a consequence of altered RAS and RAF signaling, they overexpress our old friend from way back early in the course, GLUT1. These cells have much, much higher levels of GLUT1 transporters on the plasma membrane than normal cells. Order of magnitude, okay? The reason is that, as you'll see, cancer cells have a very, very high requirement for glucose as a carbon source. They need it for metabolism. They don't need it for energy. That's a myth. They do not need glucose to come in and have it converted to ATP, like normal cells. That glucose actually goes in and it's routed because it needs the carbon. It uses the carbon for amino acid and nucleotide biosynthesis to sustain protein synthesis and cell division. Okay, so it's not really using it just for ATP. So that's the connection there. These cells have a high demand for glucose, but here's an Achilles heel. The Achilles heel is this. GLUT1 transporters are basically uniquely expressed at a very high level on these cells. So is there something, could we use this as our gateway? Could you use the high levels of GLUT1 transporters to get something into these cells that would be intrinsically cytotoxic to them? And the answer is vitamin C. Because as it turns out, okay, so here's the structure of reduced vitamin C, the structure of the oxidized, the dehydroascorbic acid, and that's what you typically find in vitamin C over the counter. It works as a reducing compound because once it gets into the cells, it's converted here to the reduced form, basically consuming glutathione, reduced glutathione. So it's the reducing activity of vitamin C that actually acts as an antioxidant. But here's what you can do. Now what you're doing is that you're giving an injection. You could never consume orally enough vitamin C tablets to get the cytotoxic effect that you're looking at here. Okay, so just to put it back on the other slide, this is the amount of vitamin C that you're having to take in to get a therapeutic outcome, okay? I don't think anybody here can eat 300 oranges in a day, okay? So we're talking a massively high dose of vitamin C. Yeah, but you can easily get that dose by an injection, okay? It's going to go in through the GLUT1 transporters. So the outset is you're not having to design inhibitors of gluten. You're not having to design something that would be a competitive inhibitor of glucose. That saves you the problem of having to compromise all the GLUT1 transporters on all of your cells, particularly your erythrocytes. You're not targeting GLUT1. You're using GLUT1 as the gateway. And the way that you can use it is these mega doses of vitamin C. Selectively, the oxidized form of vitamin C goes right through those GLUT1 transporters. It's going to get sucked into those tumor cells. Now, is it going to go into other cells? No. The reason it won't is because on your erythrocyte, you're not going to have the same numbers of GLUT transporters that you have on these cancer cells. So they don't have the ability. The other point is that the affinity for glucose is still going to be higher than the affinity for dehydroascorbic acid. You're changing the game in your favor by taking advantage of the cells that have the higher numbers of the transporters, but you can also swamp it by putting in a massive dose of vitamin C. So the vitamin C goes into the cancer cells. And here's what you're going to do. At that high concentration now, what you're doing is that you're depleting all the reduced glutathione in the cytosol. All of that reduced glutathione is now going to be used and depleted to reduce the vitamin C. Okay? So we're going to convert that to the dihydro form of ascorbic acid. Now remember, we can only regenerate reduced glutathione by using NADPH. Well, the problem is we've tapped out so much of our reduced glutathione, its level's dropping, our NADPH level is also going to drop. Okay? Now, because of the reduced glutathione, the reduced NADPH, the cell is now going to switch over from a reducing environment to an oxidizing environment. It's going to look like the inside of your ER. Well, what that's going to do is that now we're going to generate reactive oxygen species. These are going to damage the DNA in the cell. So now what you're going to do is start compromising DNA replication in these cells. Okay? Cells are basically going to have increased rates of DNA damage. They won't be able to repair it. They will die. So there's one killing effect. Here's the other killing effect, is that these increased reduced oxygen species with reduced glutathione inhibits a key enzyme in glycolysis. And that is the enzyme glyceraldehyde phosphate dehydrogenase. So in essence, what we're doing is that all of this glucose coming into these cells now is going to be blocked here at the conversion of glyceraldehyde 3-phosphate to glyceraldehyde 1,3-biphosphate. We're inhibiting a key step in the metabolism of glucose. So what that is going to do is drop the subsequent steps in glycolysis so that we're not going to make pyruvate. So what's going to happen now is we're in essence starving the cell. Okay? So what you're doing is you're causing these cells, because of the fact that they have high GLUT1 transporters, we're damaging the DNA by generating reactive oxygen species, and we're inhibiting this enzyme, which is a key step in cancer glycolysis. Normal cells don't require it nearly to this extent. So in essence, what you're doing is by using a very, very inert compound, you wouldn't even necessarily call it a drug, is that you're going to have minimal cytotoxic effects on normal cells, and you're going to selectively kill these cells, not because of the oncogenes that they're harboring, but just because they overexpress GLUT1 transporters. So this approach is actually in clinical trials now to look at treating individuals that have advanced colon cancer that's being driven by RAS or RAF oncogenes. So this is the way you have to start thinking about it. Don't just target the oncogene. Don't try and come up with cytotoxic compounds that are out there out of the blue. Go with something that is keying in on the Achilles heel of that particular cancer cell. So it's a novel approach. And so what it shows is Pauling was right just for a different reason. He was on the right track. He was well ahead of his time, but he didn't anticipate that the way that you're using vitamin C as a killing agent is to basically get it into cells because of an altered transporter. Okay? So when people ask, is this course, is this final exam cumulative? Do we have to think back to stuff from the beginning? There you go. Okay, is that far enough back? You guys remember GLUT1? Okay. Let's keep running through these oncokinases now. Because here's an interesting example. And the reason I want to talk about this is that this is an example of finding a cancer-specific kinase, a tumor-specific oncogene, where the drug works well. And what you're going to see is the problem is not with the efficacy of the drug, but with drug costs and what goes into the economics of drugs. Okay? The disease that we're looking at here is chronic myelogenous leukemia, CML, and to a lesser extent, acute myelogenous leukemia, or ALL. Immune cells are very, very susceptible to mutations that are due to chromosomal rearrangements. We're going to take a look at two. The first one is here, that basically deals with a chromosomal translocation that generates a real chimeric protein, an oncokinase. So you guys have been beat up with chimeric proteins all semester. They actually form naturally. The reason that you notice they occur disproportionately with leukemias and lymphomas is that these are all immune cell cancers. And remember that in the immune cells, as part of generating antibody diversity, your immunoglobulin genes are constantly undergoing rearrangements and shuffling. You're taking pieces of chromosomes and moving them together in order to form novel antibodies. They're literally chimeric proteins. Well, if the process doesn't go properly, there are certain chromosomal translocations that will take two different genes and put them together and make a novel protein. The one that we're going to focus is this guy up here called BCR-ABL. And what it does, it takes a normally well-regulated tyrosine kinase, which is known as ABL. Here's a close-up of that signaling pathway that I showed you guys on the very, very first class when we went into cancer. ABL is down here in the cytoplasm. Now remember, we've got the RTK pathway coming down here from the left that goes to GRAB2 sauce, and that's how we activate RAS. ABL can do the same thing. ABL is a cytosolic kinase, it's a tyrosine kinase, that when it becomes active, will also bind GRAB2, it binds sauce, and in doing so, ABL can independently activate RAS. Okay? It's a way of activating RAS from inside your cell without having to go through an extracellular growth factor pathway. Okay? ABL is normally very well regulated, well behaved. Switches on, switches off very properly so that we don't get misactivation of RAS. But that all falls apart in chronic myelogenous leukemia. And what happens is this. There's a translocation between chromosome 9 and chromosome 22. The ABL gene is down here near the distal tip of chromosome 9. It's well behaved down there. Over here on chromosome 22, we have another gene called BCR. And BCR has two different connotations. The most common one is BCR stands for breakpoint cluster region, BCR. The other name for the gene that you might find, depending on the source that you look at, is called B-cell receptor. BCR, unfortunately, we don't know a lot about it. We know it's a serine threonine kinase. It's constitutively expressed. It is always expressed in B cells. But it's inert. It really doesn't do anything deleterious, but it's just that we don't really know what it does. But what happens in this particular instance of cancer, there's a translocation that basically takes BCR and moves it over here to chromosome, it basically takes and generates a fusion where the ABL gene winds up over here on chromosome 22. And we have this little odd chromosome, which was discovered by investigators working in a laboratory in Philadelphia, so it became known as the Philadelphia chromosome. Okay? And what happens is this. It's not so much moving this little bit here of chromosome 22 to chromosome 9. That doesn't matter. We get a little bit of an elongation of chromosome 9. That doesn't matter. What matters is this alteration here. where we wind up basically losing part of chromosome 22, and now the ABL gene has moved over. Here's what's really amazing about this. It is a precise in-frame to the nucleotide fusion between the BCR coding region and the ABL coding region. And what it does is that you now generate a novel chimeric kinase. Because it's got BCR hooked up to it, it's always active. It's not properly regulated anymore. And now what it means is because of its tyrosine kinase activity, BCR-ABL will now constitutively activate RAS. Okay? It is the single most important causative oncogene that's associated with one of the most common types of leukemia that's out there, and that's chronic myelogenous leukemia. Counts for 10% of all leukemias, but as a single cause, it's one of the largest. Okay? So take a look at the numbers up there. Okay? There's about 5,000 new cases of CML diagnosed every year. So that's 5,000 new cases. There's a slight bias towards men over women. The average age of onset is mid-60s. We call that middle age. Your overall lifetime risk of getting it, 1 out of about 600. And basically what you guys can see is about 10% of these individuals that acquire it will ultimately die. Okay, but here's what you guys have. This is what's good about it. BCR-ABL is a protein you don't find anywhere else except in the CML cells. It's a novel protein. You don't find it anywhere else. Other cells have normal ABL. Other cells have normal BCR. But only these cells are expressing this chimeric protein where we've got BCR sequences right next to the ABL sequences near the catalytic site. So obviously, given the prevalence of this disease, there was a high throughput screen, and this lovely little compound over here showed to be very specific for BCR-ABL. Has minimal effects inhibiting other kinases. And it was given the name initially of imatinib. Okay, it's another tyrosine kinase inhibitor. It's sold under the generic name out there, the market name is known as Gleevec. And Gleevec was the very, very first drug that was developed to treat a tumor-specific protein. That's what made it novel, is because this drug has the potential to only target the cancer cell. You won't have any collateral damage. You won't have any side effects. So, several months ago, the Washington Post published an article that deals with the economics of Gleevec. And I want to take a couple of minutes just to show you this because it illustrates the real-world market dynamics of drugs. Okay? So, this is now updated. So you're now looking at CML is going to affect just under 5,000 people a year. It's a pretty big market. Okay? The annual price in 2001, annual price, okay, $26,000. So that basically, if you are taking Gleevec because you have CML, the treatment would run you about $2,000 a month. Not cheap, but if you want to get rid of the leukemia, that's what you go with. Okay? 2016, in 15 years, look how much the cost has gone up now. Take Gleevec now, it's up to over $120,000 a year. The only thing that keeps pace with that is UVA tuition. Okay, look at the numbers here. $1 billion a year is considered the benchmark. If you're into drug discovery, that's what you call a blockbuster. You can score $1 billion a year in marketing for your drug. That makes it a blockbuster. Initially, Gleevec was predicted, this is at the very beginning, it was predicted to top out at $100 million a year. And that was just based on the reality of the number of patients and the projected longevity of the patients. Okay? Last year, look how much Gleevec scored. Just under $5 billion. Okay, that's a 5x blockbuster. It's the single biggest drug worldwide. That means it's not only successful, it's widely used out there. Here's the initial life expectancy for people with CML, 5 to 6 years. CML patients now have normal lifespans. Okay? But you have to keep taking the drug because that's what keeps the cancer cells in check. Okay? So here's what they looked at. By 2012, this was their market. They figured they'd be treating 25 to 30,000 patients a year. By 2020, you can also see now that the number of patients going up is rising by about 5-fold. You got more people taking it because more people are living longer. But they have to keep taking the drug to basically keep the cancer in remission. Okay? Remember, you're not killing these cells. You're just stopping them from proliferating. Okay? That's an important point. They just can't proliferate. Here's where it gets really sobering. The actual cost to produce an annual supply of Gleevec, including 50% profit, markup, 50% markup, if you look at the chemicals that were needed to synthesize that, there's your annual cost. $216. So if you went in the lab and you wanted to buy the chemicals that it would take to synthesize Gleevec, $200. Okay? Annually, an annual supply, $216. This is what it's going for. That's quite a return on the investment for the pharmaceutical companies, isn't it? So here's one of the sobering things about it. Okay, first is take away the mystique, it's just chemicals, it's true. And then here's the other point. Drug prices don't really exist. List prices are nothing more than a starting point for bargaining between drug makers and the companies that provide the prescription drug benefits. This is a euphemism for saying what? Companies that provide prescription drug benefits, what do we call those? Insurance companies. So the point is this. They can charge literally what you've seen up here. They can charge whatever they feel like. It's worked out as a negotiation between the drug companies and the insurance companies, and that is what people are going to pay. You're a captive audience. If you take this drug, your leukemia will go into remission long term. The problem is the minute you stop, you can lose the remission and the cancer will come back. So to a very, very large extent, that's why you have a captive audience. It's very, very much like the drugs that we talked about earlier, the statins. The drugs that were involved, for example, in cystic fibrosis. So the thing that you guys have to appreciate when you're looking at drug development are the exorbitant costs that are out there for the public. Vastly exceed. So when you hear the argument, oh, well, it takes a large amount of money to develop the drugs. This is true. Well, it takes a large amount of money to make the drugs. No, not really. So the point is that when you're looking at the marketing and the commercial aspects of drug development, understand that the scientists, we really can't do anything about it. We can't say anything about it. But the point is it really shows you the disconnect between the ability to find a drug and what it's going to cost the end user who's out there who needs the drug to stay alive. Pretty sobering. Okay. And here's the other thing. Guess what'll happen? At some levels, it will acquire resistance. Okay? Just like this is what happens when you chase the oncogene. No matter how good the drug looks, no matter how effective it works initially, your cells will become resistant, even to Gleevec. So it turns out that individuals that are taking the drug over the long term and they're in remission, you can in fact acquire adaptive mutations that will make it resistant. In this case, the adaptive mutation, here we go again, very subtle, a single C to T transition in the second position of any threonine codon. Boy, this is great design. Threonine, if we change that C in the second position, is going to alter it to isoleucine. Isoleucine's bigger, it's hydrophobic, and it perturbs the binding site for Gleevec. So the most common form of resistance to Gleevec in CML is the acquisition of an isoleucine at that point, and that is enough to basically disturb the binding of Gleevec and prevent it from stopping proliferation. Now, do you kind of get the sense we're entering an infinite do loop here? That this is going to keep happening and happening and happening? Well, here's where we might be able to break away from what's been done in the past. And the way to do that is the following. You now know what causes the resistance. Right here. Now, do you think we have to go in and find a brand new drug from scratch, go through a screen, find something from scratch, take it through what you will see is going to take about 10 years of clinical development before it's out there? Do we literally have to start at square one to come up with a drug to deal with this? And the answer is no. Okay? What you can do is the following. And this is a paper that just came out in Nature last year. And that is, you don't start over. Instead, what you do is the following. You take your drug-resistant kinase. In this case, it would be BCR-ABL T315I. That's your target. But what you're going to do is, you don't screen it with brand new compounds. You don't start from scratch. Instead, you go back and you take just the mutant kinase. And what you're going to do is screen it with all the available drugs that are out there. Several hundred. They've already gone through clinical trials. They've all gone through phase 1, which means you don't have to worry about effects of these drugs on normal, healthy people. It saves time. And it means you don't have to redesign the wheel. And what they found was, now remember, ABL is a tyrosine kinase. So you would think, just going into this, that you might find another tyrosine kinase inhibitor that's out there that you could be able to use. And they did. It turned out that the drug that they found was this compound here called axitinib. Axitinib is already out there as a very potent chemotherapeutic drug that targets VEGF receptor, the vascular endothelial growth factor receptor. That is the growth factor in the receptor that is involved in vascularization. And the reason it is that when tumor cells become hypoxic, they're starved for glucose, they're starved for oxygen, they actually send out and release a growth factor that causes red blood cells to grow to the tumor. Okay? The cells that are growing are your normal endothelial cells. And where this particularly takes place are in kidney or renal cancers. So axitinib works as a tyrosine kinase inhibitor because it binds to the VEGF receptor. If you take a look at it structurally, it doesn't superficially resemble Gleevec. Not a lot in common. But what they found was axitinib binds with a very, very high affinity to only the T315I allele of BCR-ABL. And it does it because it binds by changing its shape. Okay, we won't get into the paper, but what it does is this compound has enough flexibility into it that it can bind very, very tightly with a very high affinity to the Gleevec-resistant form of BCR-ABL. Doesn't bind to the original BCR-ABL. So what it means is the following, is that now the approach that you would use to follow up the acquisition of Gleevec resistance is that you could switch these patients over to axitinib. And axitinib will basically knock out the mutant form of the BCR-ABL that can no longer bind to the drug in the first place. Okay? And again, by monitoring the DNA in these patients, in their blood, you can determine when in the course of Gleevec treatment that mutation arises. So you can find immediately when there are cells that have undergone that single nucleotide change, and now you can switch the treatment over to another drug. That drug has already been through clinical trials. It was approved for one cancer, so now all you have to do is switch it over and use it to treat this particular type of cancer. Okay, last point before we head off for Turkey Week. This is why targeting kinases is difficult. The reason is, as you guys know, kinases all have the same generic structure. It is incredibly difficult, even with the most specific kinase inhibitor you can find, to just target that particular kinase. Okay? So the three that are being shown here, here's Gleevec. These two guys here, Tarceva and Iressa, they target the kinase domain of HER1 and HER2. They're very specific. But what you're looking at here is that these are kinase trees. So this is looking at the functional conservation of different kinases. And here's what you notice. The bigger dot means highest affinity. Small dot means low affinity. And what this is measuring collateral damage on other kinases. And that converts to general nonspecific cytotoxicity. If you take a look here at Iressa or Tarceva, you can see up here they do a very, very good job of targeting the EGF receptor. Very high affinity. Tyrosine kinases are much easier to target than a serine-threonine kinase. And the example of that is, here's a very commonly used serine-threonine kinase inhibitor called staurosporine. Yeah, it kind of hits everything. You're not going to use staurosporine therapeutically because it's got too little specificity. So the problem that you have is that you're always trying to mitigate off-target effects by treating with a kinase inhibitor. Okay? All right. So let's call it a wrap for the rest of the week. I'll give you guys a homework assignment that'll be due a week from today. Kind of cover up this stuff. Have a good break, and see you guys next Monday, and we'll hit our home stretch.